Due to the difficulty of observing species within the marine environment, there is a lack of detailed information about many marine predators. Biologgers, like this accelerometer, afford the opportunity to follow animals without direct observation. The ability to see in greater detail what marine species are doing can give us greater insight into migration patterns, foraging habits, and reproductive strategies. Sharks are key components of marine ecosystems, and through advances in biologging technology, the movements of sharks can be examined in increasingly precise detail. There is a need to examine the movement patterns of small temperate sharks more closely, since they make up the majority of shark diversity. This is particularly the case for common species that likely play important roles in shaping their surrounding ecosystem. Port Jackson sharks, for example, are known to prey on urchins, which play key roles as ecosystem engineers on rocky reefs. The Port Jackson shark is a benthic species common to the waters of southern Australia. They have recently been found to complete migrations over 1,000 kilometers, impressive for their size. Using biologging tools such as accelerometers, we are hoping to find out more about what they do along these journeys. This representation of the three axes on the attached accelerometer have little variation when the shark is resting. When the shark swims, you can see peaks in the X and Y axes, denoting the signature for swimming. In order to find the accelerometer behavior signatures for Port Jackson sharks, we captured wild individuals from Sydney Harbor and brought them to Taronga Zoo to complete captive studies. We filmed them using high-definition video from multiple angles in the zoo enclosure, and were able to match each behavior we saw on the video to the accelerometry data. This allowed us a window into the life of a Port Jackson shark. In order to refine the accelerometer attachment method, we developed a harness that allowed for non-invasive attachment of the accelerometer tags. From these, we established the behavior signatures for resting, swimming, for vertical swimming, and for chewing. In the future, we're aiming to quantify behaviors related to reproductive success, like mating and egg laying. Armed with these behavior signatures, we can begin studies on wild individuals and facilitate insights into the behavior and movement ecology of this common species. Sharks are fundamental to marine ecosystems, and our understanding of them has increased dramatically over the last decades. In order to move toward an increasingly detailed understanding of shark movements, we want to continue examining the fine-scale behaviors of these animals.